Hello, and welcome to the second session of the Taste Masking series co-hosted by Adare Pharma Solutions and IFF Pharma Solutions. My name is Stacey McDonald-Mickey, Global Director of Marketing at Adare Pharma Solutions, and I will be moderating our session today. I'd like to introduce our speakers. Our first speaker today will be Dr. Luigi Boltri, Senior Director of Pharmaceutical Sciences at Adare Pharma Solutions. Dr. Boltri is a pharmaceutical chemist by training and completed his degree at the University of Turin. With more than 30 years' experience in the pharmaceutical industry, Dr. Boltri has held leading positions as department head and director. Dr. Boltri has extensive experience in formulation development for various dosage forms with a focus on oral delivery and bioavailability enhancement. In his current role, Dr. Boltri supports company processes leading to identify new and innovative technologies, as well as assist with our clients for product development and co-development opportunities. Our second speaker today will be Dr. Nazrin Mahmoudi, who is currently the Technical Manager at IFF Pharma Solutions. Nazrin is a member of the Application Development and Innovation team within the Pharma Solutions Business Division and a scientist and technical manager. Nazrin's role is working with pharmaceutical customers in the North American region to partner in product development. Nazrin provides technical information on application and regulatory aspects of the broad range of excipients and enables customers to troubleshoot and facilitate their formulation development processes. Nazrin has a PharmD and a PhD degrees from Medical Sciences University of Tehran, followed by postdoctoral research at Rutgers University. Nazrin has a broad range of experiences in the pharmaceutical industry, particularly in solubility enhancement, immediate and controlled release dosage forms, and taste masking of oral formulations. I would now like to hand over our webinar to Dr. Mahmoudi to start our webinar. Thank you very much.
Thank you, Stacey. We'll start with a brief recap of the first session. Next, highlighting the key product parameters that affect drug acceptability by patients. Then, an overview of technologies that improve drug product attributes along with case studies. Next, we discuss the values and importance of excipients in improving drug product acceptability and medication adherence. Then key takeaways and topics for the next webinar, which potentially will be presented by other experts and colleagues. At the end, we'll have some time for questions and answers. Now I'll turn it over to Gigi for a brief recap of our first webinar in this series. Gigi, please go ahead.
Thanks, Nazrin. Let's have a recap about our last session. In general, we discussed that poor adherence to therapy can have a negative impact on health and also higher cost for patients. At least 5 factors that may impact adherence to therapy are listed by WHO, and you can see them highlighted in this slide. Fortunately, many of the negative outcomes related to poor adherence may be prevented by understanding and addressing the related issues. Among these key factors, we discussed those critical characteristics affecting drug acceptability, including palatability and swallowability. We talk about the role of patient-centric drug design in ensuring drug acceptability and medication adherence. Patient needs and interests are different for every group of patients depending on their age, physical conditions, and disease characteristics. These needs and preferences are key to designing a patient-centric drug product. In addition to understanding the patient and disease characteristics, another important factor is drug product characteristics and target product profile in the frame of a patient-centered drug design to improve drug product acceptability and medication adherence. Nazrin now will give an overview of critical characteristics of a drug product that affect drug acceptability.
Thanks, Gigi. In a patient-centric drug design, the first step is to define a target product profile for a special group of patients. In the target product profile, the relevant characteristics of the product considering the end users, including patients, caregivers, and healthcare providers, are listed. For example, the route of administration, the dosage form, dose and dose frequency, packaging, and so on. For oral formulations, which is our focus today, drugstore ability is one of the critical attributes that influences medication adherence. For example, viscosity, mouthfeel, and dose volume for liquid formulations have impact on the swallowability. Also, for solid formulations like capsules and tablets, the size, shape, smell, taste, and mouthfeel could cause issues and difficulties in swallowing and acceptability of a medication. Factors like particle size, grittiness, and the texture of an oral formulation, its bulkiness and dose are all affecting the drug acceptability. Integrating these characteristics helps achieving a drug product suitable for pediatrics, geriatrics, or any other special group of patients that present particular complexity with respect to medication adherence. Fortunately, there are several approaches, technologies, and functional excipients available and are being used by pharma industries to deliver patient-friendly drug products. That we'll discuss few of them in our webinar today. I will now turn it over to Gigi to present some of these useful technologies.
Thanks, Nashreen. We are now stepping into a deeper dive in those technologies and formulation strategies able to address the challenge of a patient-centered formulation, with a focus on the more challenging patient segment, for sure children, but we should not forget elderly and the need of improving their quality of life by facilitating the administration of drugs, sometimes multiple drugs. First, we'll have a look at the Adare technology platform, dedicated to the development of small taste-masked particles, including some case studies of successfully developed drugs. Then we'll move to those technology and formulation approaches intended to complete the development journey by offering a more complete patient-centric solution. Finally, just a glance at a new approach to drug administration that in a certain sense challenges some pillars of the formulation standards. Let's start with analyzing encapsulation as a tool for addressing the bad taste that unfortunately characterize the majority of drugs. Adare has two different technologies leading to small particles, let's say starting from 100 microns, able to mask the taste by processing the API with some functional excipient. Besides an effective taste masking, small particle size is required to minimize grittiness and sandy perception, being another critical factor impacting medicine acceptability and adherence to the therapy. Optimum is the first one I'm going to present. Optimum is one of the applications developed by Odps Biosciences, recently implemented and integrated within the Adare organization. Main features of this technology are particle size and particle shape uniformity, as you can clearly see in the pictures where particles generated by conventional processes are compared with particles generated using the Optimum process. The technology is quite flexible, being able to utilize several and diverse functional excipients to generate matrix and core shell particles. Matrix particles have an homogeneous composition, and the API is thoroughly dispersed within the particle bulk. On the other hand, core shell particles have a core, usually comprising the drug, surrounded by a functional shell. Basically, it's a two-compartment concentric particle, or simply a microcapsule. Let's have a look to how these particles are generated. Basically, Optimum is a melt spray congealing process, more precisely a melt drip congealing process. Therefore, this technology makes use of a nozzle, but this nozzle is designed to overcome some of the major limitations of a dripping process. The particle size achievable with a vibratory nozzle is about 2 times, sometimes more, larger than the diameter of a nozzle orifice. This means that to create a 100 micron droplet, a nozzle orifice of 50 micron or less should be used, unavoidably leading to severe limitation of productivity and limiting also accessibility to low viscosity fluids, as well as increasing the risk of nozzle clogging. optimal nozzle, besides being equipped with a frequency generator able to precisely cut the fluid jet stream, leverage the effect of an annular stream that narrows and accelerates the nozzle jet to smaller than the orifice, which allow for small droplet size and minor impact on process output. Same nozzle configuration can be used for generating core shell particles. The only difference is the presence of a concentric nozzle, basically adding another fluid layer to enable the generation of a core shell structure. This allows for a single-step coating mechanism while maintaining a small size. By tuning processing variables, we can precisely control particle size, but also the core shell ratio, as you can see in the slide. I'm presenting now a selection of case studies where Optimum has been successfully applied. Here you have some examples and case studies of both the particle configuration Optimum can design, matrix and core shell. Highly bitter or bad-tasting molecules, such as cetirizine, prednisone, and ritonavir, can be processed using the Optimum technology, leading to tasteless particles having a diameter in the range of 100 to 200 microns. And looking at the dissolution profile, it is evident that Optimum technology is able to address the most challenging formulation target, balancing taste masking and immediate release profile. As you can see, taste masking is ensured by minimizing the release in the first minutes after the administration, when the dosage form is in the mouth. Drug is then quickly released in the stomach in order to ensure bioequivalence with the reference product, usually represented by an immediate release dosage form. Obviously, also sustained or delayed release profiles can be developed by selecting the appropriate functional excipients. Let's spend now a few minutes on microcaps. Microcaps is the technology on which Adare was originally created more than 50 years ago. It is therefore a consolidated and commercially validated technology, and microcaps products are present in the main regulated countries. As per the core shell application of the Optimal technology, microcaps is able to apply a uniform coating on a particle. Particle to be coated can be plain API, crystalline powder, or an intermediate such as granule or pellets. Co-absorption is a physical phenomena leveraged for the application of a continuous coating around the particle. Co-absorption can be described as the coexistence of three different not mixable phases in the system. On the left side, you can see some picture representing the sequential steps of the microcaps process. In the first picture on the top, you can see the core particle representing the first phase suspended in a liquid vehicle. The liquid vehicle is a solution of the coating polymer. During the process, we can induce the co-absorption phenomenon, basically a salt-gel transition. And what you see in the background are the gel globules having a high concentration of the coating polymer, representing the second not mixable phase. The third phase is not visible and is represented by the residual solution of the coating polymer. Looking at the picture on the middle, you can see that the gel phase starts enveloping the cores, and even at the initial phase of the deposition process, a tiny but continuous layer can be already seen. The driver for generating the coating is mainly thermodynamic, which is with the objective of reducing the interfacial free energy. The picture at the bottom represents the completion of the polymer deposition, and it is possible to appreciate that the deposition is quantitative. There is no more polymer in the liquid media. The peculiar boat shape of the membrane is due to the hydrodynamic of the process, where steering is used to keep particles in suspension and minimize or control aggregation. Important to note that here the coating is in the swollen state, and once the residual liquid media is removed by filtering and drying, the coating becomes much thinner, robust, and rigid. On the right side, you can see the list of the major and distinctive advantages of microencapsulation versus alternative and traditional coating processes. The particle size is small, so the final particle size can be down to 150 to 100 micron at the industrial scale. It is a highly efficient coating. Usually 10-20% of coating are enough. And I don't remember on the cases above 25-30%. This can reduce bulkiness for high-strength medicines. Processing time is relatively short, one shift, one batch, and another has high manufacturing capacity. For instance, in the case of potassium chloride microcaps, last year we produced more than 800 tons of processed material. It's also able to manage not ideal particle shape thanks to the flexibility of the coating during the process, and we can also process micronized material using controlled aggregation, and you will see an example later on. And now I'm going to present one specific case studies where microcaps has been successfully applied, leading to a successful commercial product. Hexafenadine is quite a bitter drug, and Sanofi was looking for a pediatric formulation for the Japanese market in form of powder, able to mask the taste, but most important for Japanese children, particle size had to be quite small, below 200 microns, in order to minimize grittiness and particle perception. This is an example of designing a product profile not only taking into account the patient, children in this case, but also some cultural aspects, such as an enhanced accessibility to grittiness and sandy effect. Moreover, the product was required to be bioequivalent to an immediate release reference tablet, and the possibility to administer product as is on the go without water or pre-suspended in a small amount of liquid was also required. The solution was to combine microcap in order to generate a small taste-masking particle with Liquidator, basically an excipient blend able to facilitate administration without water, but also suitable for generating temporary suspension. As you can see, challenging and complex formulation targets usually require the synergistic combination of more technologies. Here you have some pictures representing the drug particles in the range of 20-30 microns crystals and the corresponding microcapsules generated by a single step of granulation and encapsulation. These provide evidence that the microencapsulation process can manage also fine particles, and the final outcome can be still targeted in the 150 to 100 micron range. Despite the small particle size and large surface area, the amount of coating required is quite low, in the range of 15%. The coating deposition is confirmed to be quantitative, therefore highly efficient. Particle size is in a range where grittiness perception is minimized, and the solution profile is superimposable to the profile of an immediate release form. Even if the drug release in the first minutes after the administration is much lower to ensure taste masking. Last but not least, Adare took care of all the development work, from prototype design to commercialization. The product has been launched on the Japanese market a few years ago, and we have been also able to add valuable intellectual property to the product concept. Let's move now to a handful of technologies and formulation approaches intended to complete the development journey by offering a more complete patient-centric solution platform. Patient-centric solutions are required for all the patient population, but there are patient segments requiring special attention. Children, elderly, and disfaged patients are the most representative example of the need of including acceptability as key factor in any new target product profile. This is the reason it is fundamental to develop also those technologies and formulation capabilities able to transform an enabled intermediate, such as an optimum or microcaps taste-masked particles, in an acceptable final dosage form, or medicine if you prefer. Here you have represented a span of possible dosage forms designed to accommodate optimum and microcaps-enabled particles. To name a few, temporary suspension, dispersible sachet or tablet and parvolet, and novel formulation concept are useful for administering therapies to patients who may find taking conventional dosage forms to be difficult to swallow, We'll talk later about Parvolet. Let's start with Liquidator, an excipient blend designed to be combined with small taste-master particles to generate temporary suspension, sprinkle, or even on-the-go dosing. But taste-master particles originated with optimum and microcaps technologies can be also incorporated in an ODT, an orally disintegrated tablets. And to minimize the risk of breaking particles during the tableting process, jeopardizing the taste masking properties, a specific ODT composition and manufacturing process named AdvaTab was developed internally, leading to an ODT able to load a significant amount of active and disgregate quickly in the mouth. Finally, just a glance at a new approach to drug administration that in a certain sense challenges some pillars of formulation standards. Patient adherence can be improved through the development of innovative dosage technology, especially for patients who struggle to ingest standard oral dosage form. For many valid reasons, most drugs are currently administered using solid forms, such as tablets and capsules. However, the deglutition process is a complex mechanism, comprising a preparatory phase in which the food bolus is chewed and wetted with saliva to define texture and rheology, so to activate the natural deglutition reflex. Therefore, you should assume that human beings are not designed for swallowing solid objects, such as capsule and tablets. Parvolet is a solid formulation, tablet or granules, as you can see, but able to turn into a soft food by simply adding a few milliliters of water and waiting for 20-30 seconds. It is important to note that no other human intervention is needed after water is applied, which further reduces the risk of human error or misuse. This delivery design represents a major paradigm shift in oral drug administration, but the video is better than 1,000 words. As you will see in the video, the administration procedure is quite simple, and by adding a little amount of water to a Parvolet granule or a Parvolet tablet, it is quickly converted in a soft food bolus, easy to swallow. Important to note that besides improving product acceptability, Parvolet overcomes some mispractices, such as crushing tablets or open capsule, mix the powder with food or naturally thick food vehicles, such as yogurt, jam, or pudding, that could impact on stability and the solution properties of the API. So let's have a look to the video, and then Nasrin will provide a deeper dive in how the appropriate use of selected excipient can help in improving patient acceptability.
Thank you, Gigi, for highlighting interesting technologies and case studies to improve drug product acceptability. I'm going to start with oral drug product acceptability and then talk about application of excipients in improving drug acceptability in patient-friendly dosage forms, including orally disintegrating tablets or ODT, chewable tablets, conventional tablets, and suspension formulations. The importance of swallowability and palatability as critical characteristics of oral medicines, useful guidelines, and requirements to design an acceptable drug product to improve medication adherence have been discussed, and it was shown that a number of tools and techniques are available for pharma industry to overcome challenges with drug acceptability. Let's see how using some functional excipients can improve drug product characteristics such as taste, texture, and the image size. Major techniques for improving drug acceptability, from simple methods to more complicated ones, rely on using functional excipients. Developing a palatable formulation could be very challenging, particularly for dosage forms with long residence time in the mouth, like orally dispersible dosage forms, chewable tablets, and liquid formulation. These types of products will require technologies that can provide sustained taste masking compared to a dosage form like tablet that is swallowed intact or unbroken. We often need to use a combination of strategies, and no single technology can work for all cases. Most taste masking techniques require high-quality, multifunctional, and safe excipients, which I'll review some of them today. In terms of techniques, polymer coating is one of the most efficient technologies used in taste masking. Ethyl cellulose and acrylates are among the most commonly used polymers for sustained release coating and taste masking applications. Ethyl is the trademark name of ethyl cellulose, and we'll take a look at its properties and its application next. Ethyl cellulose, with the trademark name of Ethyl, has been manufactured in Midland, Michigan for more than 80 years from cellulose. Through chemical reaction, cellulose is ethylated and converted to ethyl cellulose under tight control conditions, followed by purification processes. Viscosity of available grades of ethyl is shown in this second table. In IFS, we have a tight control over the degree of ethoxy substitution and solution viscosity. These two properties are the most critical variables in the final properties of any ethyl grade. Ethyl polymers are safe and considered GRAS by FDA, can be used in pharmaceutical, nutraceutical, cosmetic, and personal care products. One of the important properties is their solubility. They are not soluble in water, but are soluble in a variety of organic solvents such as methanol, ethanol, and also are soluble in natural oils. They can be used in different processes such as direct compression, wet granulation, hot melt extrusions, microencapsulation, and polymer coating. Ethyl films are strong and at the same time stretchable and flexible even at low temperature. The application of ethyl in pharmaceuticals are mostly related to barrier membrane film coating and its lack of water solubility. Ethyl can be sprayed from organic solution, aqueous dispersion, or even used as dry powder for a solvent-free coating. Drop release can be modulated by polymer viscosity or coating weight gain, which is much easier. The graph on the left shows that by increasing the viscosity of polymer, we can slow down the release. And the graph on the right shows that drop release can be reduced by increasing the coating weight gain. Another grade of ethyl is ethyl Hp that is designed for dry coating application and has macronized particles. The HP in the name refers to high productivity. Using ethyl Hp, coating process would be much faster and more efficient, potentially 3 to 4 times increase in productivity. So multiple options are available for application of ethyl and tailoring the drug release. For taste masking application, ethyl cellulose can be coated on different substrates such as tablets or multiparticulates. When coating with drug particles, the water-insoluble nature of the polymer delays the drug release, creates a lag, and prevents immediate dissolution and release of the drug in mouth. As a result, we don't feel the taste of the drug. Quality. Quality of ethyl in any application is critical for a robust performance and reproducible result. Unreacted cellulose fibers from ethylation reaction during manufacturing could be problematic. Ethyl has lower insoluble fiber content as compared to other ethyl cellulose brand in the market, 15 ppm in ethyl versus about 1200 ppm in the other brand of ethyl cellulose. High level of insoluble fibers would result in producing films with poor quality, which can cause premature drug release adverse effects or the need of, for excessive coating rates to obtain reproducible results. We've done a quick experiment to show the impact of insoluble fibers. In a low concentration solution of cellulose, fibers are optically transparent to the naked eye and don't give any indication of their presence. But under polarized light, the suspended fibers in high concentration solutions scatter light and make the solution look turbid. This picture shows 15% solution of cellulose in organic solvent under polarized light. Ethyl solution is on the left, and the solution of the other brand of cellulose is on the right. The right sample is more turbid, indicating higher level of insoluble fibers. The respective films from each solution are shown here. The other brand of cellulose scatters more light as compared to ethyl film, indicating imperfection and defects. A defect-free film will avoid the most common failure in barrier membrane coating, like film ruptures and dose dumping, as mentioned. Despite the insolubility of cellulose in water, there are commercial versions of pre-formulated aqueous dispersions are available and used quite widely for ethyl coating. For example, show release from ColorCon and Aquacode Ecb from IFF. I've selected an example of aqueous application of cellulose using Aquacode Ecb to discuss today. Aquacode is a dispersion of cellulose with 30% solid and require addition of a plasticizer before application. It is compatible with different hydrophilic and hydrophobic plasticizers. Let's take a look at the role of plasticizers as one of the critical formulation parameters when applying Aquacode Ecb coating. Cellulose has a glass transition temperature, or TG, of 91 degrees centigrade. It is very rigid at lower temperatures without plasticizers. With the addition of about 25% plasticizer to the coating formulation, the TG reduces to 35 to 40 degrees centigrade. At this temperature, the solid particles can deform to coalesce and form a continuous and ductile film surrounding the substrate. At lower concentration of plasticizer, the polymer cannot form a continuous film, and the film would be brittle. We can see cracks on the coated pellets in this same image, but at about 25% plasticizer, a continuous layer of the film without cracks is formed as shown in this image on the right. Drug release from these pellets are robust, predictable, less variable, and slower than the release from the pellets with the defective film. So it is important to add sufficient amount of plasticizer in the coating formulation and make sure the coating temperature is above the glass transition temperature of the coating. The hydrophobicity of cellulose can be reduced by increasing its porosity by adding suitable pore-formers. Pore-former types and level, plasticizer types and level. In addition, percent of coating or weight gain are critical formulation parameters that must be optimized to achieve desired drug release. In addition, to have a stable and robust formulation, a complete film coalescence and curing is required. Now let's talk about application of Aquacode Ecb. One of the common application is taste masking of multiparticulates for use in orally disintegrating tablets, in suspension, and other dosage forms. Let's start with ODT. A quick introduction. ODT is a patient-friendly drug delivery system that are popular with hospitals and healthcare providers because of improved patient compliance, particularly for patients with difficulty in swallowing, which is estimated to be 25% of hospitalized patients. It is becoming more and more popular among patients and healthcare providers because of the ease of use and accurate dosing. The global market for ODT was predicted to increase at a compound annual growth rate of 11.5% in 2017, which means from about $11 billion in 2017 would grow to $27 billion by the end of 2025. I'm going to show you a case study where we used Aquacode Ecb with a hydrophilic plasticizer to achieve taste masking and improve acceptability in an ODT formulation. In this case study, paracetamol was selected as a mother drug and coated with Aquacode Ecb in a fluid bed using bottom spray method. Metocell VLV, which is a very low viscosity grade of Hpmc, was used as pore-former, and THC as plasticizer, as shown in this table. The graph shows the dissolution of granules coated without pore-former at 3 weight gain, 5, 10, and 15%, and cured for 4 hours in an oven. It is shown that the drug release rate decreases as coating level increases from 5 to 15%. Without pore-former, it's more challenging to modulate drug release accurately and flexibly. The permeability of cellulose film can be increased by incorporating a pore-former, as mentioned. We can adjust the drug release by coating weight gain and pore-former levels. These two dissolution profiles on the center and on the right show the impact of pore-former and coating weight gain on drug release. Metocell VLV was evaluated at 20 and 10% with respect to the polymer as pore-former. It was found that the 20% pore-former level with 20% weight gain hit the target delay release profile while providing a complete drug release. These are SCM images of uncoated and coated samples. Uncoated samples on the top have rough surfaces which were nicely covered with a continuous and uniform film of epicellulose. Coated granules without pore-formers shown in the middle row and with the pore-former shown at the bottom. Both have smooth surfaces as shown in these images. At next, the coated paracetamol granules were evaluated in an ODT formulation. The coated granules and the functional excipients shown in this table were incorporated into the ODT formulation using a simple direct compression method. The resulted paracetamol ODTs were evaluated for physical properties, disintegration time, drug dissolution, and taste masking performance. The study showed that the tablets had a short disintegration time of less than 30 seconds, full immediate release, and acceptable taste based on evaluation by a tasting panel. The evaluation confirmed that the coated granules were qualified for application in ODT formulation. In addition to taste masking, excipients can help to improve drug products' texture, mouthfeel, and swallowability. Avistol CE15 is one of the co-processed excipients designed for chewable tablets, which we discuss next. Microcrystalline Cellulose, or MCC, with the brand name of Avistol from IFF, have been continuously improved to address various formulation challenges. Avistol C15 is a co-processed MCC and 15% guar gum. It is used in chewable tablets to improve sensory characteristics and organoleptic properties of the tablet. It helps to lessen grittiness, to reduce tooth packing, and minimizes chalkiness. It imparts a creamy mouthfeel. It has great compactability and can decrease friability at a low level of use. I'll have a brief introduction on chewable tablets and then review the case studies that show some of the application of Avistol C15 in chewable tablets. Chewable tablets are another patient-friendly dosage form. They are intended to be chewed and then swallowed rather than swallowed whole. So they should be designed to be palatable and be easily chewed and swallowed. Should be safe and easy to use in a diverse patient population of, pediatrics, adults, or elderly patients who are unable or reluctant to swallow intact tablets due to the size of the tablet or difficulty with swallowing. Chewable tablets are available for many over-the-counter and prescription and drug products. It is growing fast. For example, the probiotics market is expected to grow at a CAGR of 6.7% to reach more than $76 billion in 2027. Some adverse effects are reported for chewable tablets, including gastrointestinal obstruction as a result of patients swallowing whole or incompletely chewed tablets. The other reported adverse effect is tooth damage or denture breakage as a result of excessive tablet hardness, and other reports, related to esophageal irritation. Due to the reported adverse events, FDA published a guidance in 2018 that describes the critical quality attributes for chewable tablets like hardness, disintegration time, and dissolution. In addition to these critical parameters, it is important to pay attention to tablet size, thickness, and friability, as well as taste, which may impact the ability or willingness of a patient to chew the chewable tablet. This means that a patient may swallow whole tablet rather than chew a bad-tasting tablet. Let's move on to case studies now. In these three examples, to improve properties of chewable tablets, we used Avistol CE15. In the first two cases, chewable paracetamol and chewable aspirin tablets for adults had chalky mouthfeel and left chalky residues with high grittiness. Tablets were reformulated and 10% Avistol C15 was added to the powder balance to replace the same amount of mannitol. This small modification greatly improved the tablet mouthfeel, reduced grittiness and chalkiness, tooth packing, and overall it was perceived a better taste during evaluation. In the third case study, the problem was extreme hardness and high friability of chewable antacid tablets. To optimize the hardness and reduce the friability, 9% Avistol CE was added, which easily solved the problem. Another example for improving drug acceptability using high functionality excipient is Avistol Dg. Avistol Dg is a co-processed excipient containing 75% microcrystalline cellulose and 25% anhydrous dibasic calcium phosphate that produces high-quality tablets, particularly in dry granulation, where we need to use material with high compactability and high revocability. One of the commercial ginkgo formulations contains approximately 11% active and 89% of excipients per tablet. We reformulated the product and incorporated Avistol Dg which reduced the amount of excipients to 67% and reduced the number of excipients from 6 to 3 as listed here. As a result, a much smaller tablet, almost half of the original size, with excellent physical property and hardness was obtained using a much simpler and more economically manufacturing process. Liquid formulations such as solution and suspensions or reconstitutable powder for liquid formulations are patient-friendly dosage forms, especially for use in pediatrics. I'll discuss the application of colloidal Avistol in suspension next. Colloidal Avistols are co-processed microcrystalline cellulose and sodium carboxymethylcellulose. Due to functional synergy, colloidal Avistols have improved suspending property, higher thixotropy and gel strength, and improved stabilization properties. Let's quickly review critical attributes of suspensions before the case study. A suspension often contains multiple ingredients to address critical attributes such as taste, mouthfeel, drug stability, viscosity, pourability, and content uniformity. One of the critical ingredients in the suspension formulation is suspending agent. Colloidal Avistol are often used as main suspending agents and also as a texture modifier. And now let's review a case study. In this case study, a paracetamol sachet formulation for use as reconstitutable suspension was prepared. At first, sugar sphere pellets were coated by a layer of paracetamol, followed by Aquacode Ecb coating for taste masking. Next, the coated pellets blended with other ingredients. Colloidal Avistol CL611 was used as the suspending agent and mouthfeel modifier. For evaluation, 1 tablespoon water was added to the powder and agitated before testing for palatability. Taste evaluation was done by a taste panel of 5 people. Paracetamol uncoated pellets received a score of 0, indicating the most bitter sample, and pellets coated with Aquacode received a score of 4.5, which was perceived better when tested in the suspension that was described as a creamy and easy to swallow formulation with a score of 5. With that, I would like to thank you for your attention, and we'll turn it over to Stacey to tell us about the next webinar topics in this series. Thank you.
Yes, hi. Thank you to our speakers today for the webinar, and I'm going to kick off with our questions from today's webinar. The first question is it possible to use Aquacode Ecb for taste masking through wet granulation?
Thanks, Stacey. Good question. Yes, taste masking can be done by using Aquacode Ecb as a granulating liquid in a fluid bed granulator or in a high shear granulator. In one study, we used a macronized API which had a neutral form and granulated with both processes. The high shear showed better taste masking potential with a lower weight gain compared to the granulation in the fluid bed. In that study, to further improve the taste of the final granules, the API and a mixture of MCC and lactose all together were granulated and compared to granulating the pure API. The taste was more improved by granulating the premix of API as compared to the pure API. Overall, both methods may provide efficient taste masking while maintaining immediate release profile of the drug, which is critical for an IR dosage form.
Great, thank you. Our next question is, Optimum uses melted excipients and temperature is a driver for microcaps to induce coacervation and activate the coating process. What is the impact on thermolabile drugs?
That's a very important question. Yeah, it's true. It's true that there is a certain use of temperature for both the processes. It is important to underline that both the processes can be managed in a way that the inert atmosphere can be created in order to prevent and minimize the impact of what is at the end responsible for degradation in thermolabile drugs, which is a combination of humidity, oxygen, and temperature. So, and making use of lipophilic excipient in the case of Optimum, and also organic anhydrous solvent and liquid media for the microcaps, then we can minimize and prevent the impact of temperature in sensitive drugs by preventing hydrolysis and oxidation. Just to give an example, we have been able to process biological material with both the technologies without experiencing a specific or significant degradation.
Great, thank you. And our third question, are there any water-soluble polymers other than ethocell that can be used for taste masking, and do they need pore former for their application?
Sure. There are some vinyl polymers like Aquacode Ecb, that have been used for taste masking. Also from methacrylate polymers, different grades of uterogate have been used. In addition, ceramic cellulose acetate can be used for taste masking. And in terms of pore formers, when we use water insoluble polymers for taste masking, often we need to add a water-soluble polymer as pore former to ensure a fast drug release after an initial lag or a delay in release of the drug in mouth. So by adjusting the ratio of the two polymers, we can achieve a lag time that is needed for taste masking, followed by a fast drug release.
Great, thank you. Our next question, you said current practices such as sprinkling on food can have an impact on stability and dissolution. Could you please provide more granularity on that?
Yeah, I can take this question. Yeah, it's true. There is a growing attention by the regulatory authorities to what can happen to a product, to a pharmaceutical product, a medicine, when it's in contact with a vehicle, like, a soft food. And what can happen in the light of possible interaction and incompatibility and then degradation or alteration of the coating or drug delivery mechanisms that are controlling the rate of availability of the drug in the body, then jeopardizing the control of the dissolution rate and the rate of absorption. This can lead easily, especially for once-a-day formulations or controlled release product, to a higher exposure and also of the drug in the body and absorption, and then leading to possible, unwanted side effect or even toxicity. Because basically by losing control of the mechanism of release control, you are basically exposing and providing the entire dose to the patient that was intended to be diluted over the day and controlled, for a longer period of time. And all these aspects, are then basically, we have some regulatory guidances now, quite recent, a couple of years ago, that are now ruling the application of products that are intended for sprinkle, and then, the bar in terms of demonstrating that the process of sprinkle is safe is quite high now. And the regulatory agencies, are looking to demonstrate, with experimental data, there is no impact on the biopharmaceutical properties of the medicine, and then there is no impact on the quality and the safety of the administration of medicine.
Thank you, Gigi. And our next question is, can Hpmc be used for taste masking coating?
Can Hpmc be used for taste masking coating? Yes, it's been used for taste masking. An advantage of water-soluble polymers like Hpmc and HPC is that they only delay the drug release for a short period of time and do not delay the drug release afterwards. But on the other hand, they provide only short taste masking periods, relatively speaking, and efficiency may not be the same as those that are insoluble in saliva.
Great, thank you. And for our next question, what are the methods to improve texture or mouthfeel of a drug product by excipient?
A drug product that leaves gritty or rough particles on tongue and imparts dry feeling in mouth is less acceptable by patients. Water-insoluble ingredients, both excipients and drugs, can cause this unpleasant grittiness and texture in mouth. Therefore, reducing the particle size of the API and excipients can improve texture. For instance, a grade of MCC, Avistol pH 105, with a mean particle size of 7 micron, performs better with regard to texture compared to another grade of MCC, Avistol pH 102, for example, with a mean particle size of 100 micron. As mentioned, there are some excipients like Avistol Dg that are available and are designed to improve texture of chewable tablets. Sometimes we can improve texture of ODT formulations by including soluble excipients or increasing the ratio of soluble excipients if possible. In some cases, we can improve the mouthfeel by adding mannitol or other sugar alcohol, which has negative heat-up solution to produce cooling and sweet taste sensation. These were some examples to improve the texture of solid dosage forms. For liquid formulations, increasing viscosity by adding excipients like xanthan gum or texture cells, it is like sodium carboxymethyl cellulose, we can improve organoleptic properties and mouthfeel.
Great, thank you, Nazar. And I believe we have time for one more question, and that will be, you mentioned that sustained profiles can be achieved with small particles down to 100 microns. Could you please elaborate on this, considering such a large surface area?
Yes, that looks a challenge and is a challenge because when you generate small particle down to 100 microns, then the surface area that's controlled in terms of controlling the dissolution and the permeation rate of the molecule through a membrane is huge. It's used in a matter of square meters per gram. And especially in the case the compound you have to control the release is highly soluble, then it's basically impossible to use a standard coating approach with polymers that have anyhow a certain permeability to the API, to the compound. And then you need to use more lipophilic material, either matrix, but more appropriate as a coating. And this is one of the advantages of having, the optimum technology in which you can play with low melting lipids and lipophilic material that can be also combined with other, polymers in order to have, a very good and sustained release up to, 12 hours, even with, a 100 micron, 200 micron particle, incorporating a highly soluble drug. And that, is a unique application of the Optifone, platform.
Great. Thank you, Gigi. At this point, I'm going to conclude our webinar. Thank you, everyone, for joining us today and for our speakers. Please join us next time. We'll be having our third series of the webinars in early June that will be focused on flavors, taste modulation, and regulatory challenges. We'll also be sending out the on-demand version of the webinar and the slides from today's presentation out to everybody registered within 24 hours. Thank you again, everybody. Thank you for joining our webinar.